Hello everyone, my name is Magda Mustile and I am a PhD student from the University of Stirling. Today I will present an investigation on motor imagery and action observation. Motor imagery and action observation share similar features with action execution, and they have been largely used in the recovery of motor impairments. However, there is a lack of evidence on how these techniques are of benefit in the treatment of motor disorders and whether they involve same motor processes associated with movement execution. Here we compared cortical motor processes during motor imagery, action observation, and actual execution of working in order to provide new evidence for the rehabilitation of gait disorders. We recorded the brain activity with a mobile EEG while participants were actually walking, observing a model walking and imagining to walk. We found that only motor imagery of walking but not action observation shows similar brain activity compared to action execution. This might have critical implications for the rehabilitation of gait disorders.
Hi, I'm Lauren, and my poster is called 3D printed tissue mimicking in cell-compatible hydrogels for ultrasound and microbubble research. So some background to our work is that a new in vitro experimental setup that involves 3D printed tissue mimicking BIMA hydrogels has been created to investigate ultrasound and microbubble mediated drug delivery. And we think that incorporating cells would improve this even further. So our research questions are: 1, can the BIMA hydrogels be easily modified to better support cell adhesion and proliferation? And if so, which materials are best and in which proportions should they be included? So we did this by seeding smooth muscle cells onto the surface of some of the different combinations of materials, including acrylic acid, polyvinyl alcohol, and gelma. And then we assessed cell growth and survival in these new materials in comparison with a tissue culture plastic control. So these are just some of our results here.
Hello everyone. The topic of my presentation this time is low-cost photoacoustic computed tomography system. This is an adaptive back projection reconstruction. Now, a background.
Hi, I'm Amy and my poster is on the assessment of the Lungman anthropomorphic phantom. As you can see here, this is Lungman, our anthropomorphic phantom, and the research question we wanted to answer was, is Lungman representative of our local patient cohort? So in order to determine this, we exposed Lungman on 2 different X-ray units in Ninewells Hospital according to the local protocols for each of those rooms. We then carried out patient dose audits for both of these units so that we could see how our exposures of Lungman compared to the exposures of our local patient cohort. What you can see here is one of the distributions from our patient dose audits for one of the units, and the purple line shows the cap that Lungman received as part of its exposure. And if you want to know a bit more about what this means, please come and visit my poster at P16.
Hi everyone, welcome to my lightning talk, and my poster today is going to be on developing new biomarkers for imaging oligodendrocyte function in vivo, and I'm Robert Shaw. So the research question we had is S1P5 expressed at the protein level in oligodendrocytes, and if so, does our ligand selectively bind to S1P5 in rat brain tissue? The method we used to do this was immunofluorescence in order to run co-stains with different cellular markers to pinpoint S1P5's expression on oligodendrocytes, and to use tritiated ligand binding assays to assess if our ligand selectively binds to S1P5, much like a key in a lock. Little sneak peek of the findings, we managed to develop a functional protocol for staining S1P5 and co-localized S1P5 in whole adult rat brain tissue sections along with other cellular markers. However, our ligand did seem to display non-specific binding in rat brain tissue homogenate. But come over to the poster for a bit more information later in the afternoon. Thank you.
Hello everyone, my name is Patricia Foya and I'm going to present the study done to establish the parameter space of ultrasound imaging-based detection of tissue stiffness variation. Our main motivation for the study is pancreatic cancer, for which, using ultrasound imaging, tissue stiffness can be used as a biomarker for early detection. In order to determine the efficiency of this method, we need to determine what the smallest stiffness difference and smallest tumor size we can detect are. We first employ OnScale, an ultrasound simulation software in which we create a simple tissue model which is then interrogated. We also create tissue mimicking materials using polyvinyl alcohol. These samples were tested mechanically and are currently being tested acoustically. Our results allow us to determine the smallest stiffness difference between the tissue and the tumor and to determine the smallest tumor size. Whether this enables earlier detection will be discussed on the 17th.
I am Nafisa from University of Aberdeen. The topic of my research is association of regional cortical complexity with fluid cognition and life course factors. The brain complexity had shown to be strongly correlated with cognition and education. Here we investigated the association of fluid cognition and modifiable life course factors with regional cortical ribbon complexity in middle to older age adults. We obtained T1 weighted imaging data from UK Biobank imaging study. Box counting method was applied to measure the cortical ribbon complexity, which showed significant positive correlation with fluid cognition in all regions. This association was independent of age and sex and other life course factors at hemispheric level and in several lobes. Our large-scale studies support the notion that income and healthy lifestyle may be a protective measure against cognitive decline. For more information, please visit my poster P19. Thanks.
Distribution of muscle stiffness plays an important role for joint stability during operation of functional movements. Ultrasound shear wave elastography provides a reliable tool for quantifying tissue stiffness. However, applying this method to examine elastic properties of human muscles have been limited. Therefore, objective of this study to evaluate muscle stiffness using ultrasound shear wave elastography. For preliminary work, landmark of flexor digitorum superficialis, the key muscle to control movement of the hand is determined to use as the ultrasound scanning point. Muscle stiffness of different intensity of muscle contraction is evaluated using a veloconic research system. The result of this study considered as a promising method and provide a basic information for further study about mechanical properties of muscle of upper limb involving the functional movement of the joints.
Hello, my name is Tianyu Zhang. My poster number is P22. In different timelines of wound healing, vascular changes of the skin play a vital role. And the OCT angiography is an ideal technique for this situation. Hence, this is my research using a swipe source OCT system to monitor wound healing process. We image 8 mice with acute wounds, 8 with chronic wounds. In the post-processing, we utilized several algorithms to achieve the best presentation of the vascular structure. We generated ENFAS structural and vascular images and epidermis layer thickness mapping, which demonstrated that our system can provide abundant information within different phases of wound healing. Thank you.
Hello everyone, my name is Yilong Zhang from University of Dundee. My poster number is 24. In this study, the effect of cell matrix interaction on the viscoelastic property of tissue engineering stromal model during a continuous culture period were explored. In the method part, I introduced hydrogel operation, air pulse-based surface acoustic wave OCE method, and plate compression rim hold relaxation test. In the results, the elastic modulus and the viscosity of the cell-scented hydrogels continuously increased over 7 days. The OCE results had a high correlation coefficient with the compression relaxation test. Come to find more details in my poster section. Thank you.
Hello, I'm Catherine, and my poster number P25 is about our assessment of MR imaging quality with an MR-guided focused ultrasound installation. The University of Dundee has recently installed an Exablate Neuro MR-guided focused ultrasound system to its 3 Tesla Siemens PrismaFit for use in non-invasive neurosurgery. Our aim was to investigate how the installation of this piece of equipment might affect the MR image quality. We performed QA before and after the installation to assess any differences, and images were required of a healthy volunteer. The helmet doesn't fit in the head coil, so the integral body coil must be used, which significantly impacts the image quality, as you can see here. We experimented with fMRI using the body coil with some surprising results. The blue here is bold fMRI signal detected by the head coil. The red is what we unexpectedly detected using the body coil. If you would like to find out more, please visit my poster number P25.
Hello, I'm Chris Taylor, and my poster is about the characterization of a diffusion phantom used for diffusion MRI QA. NHS Tayside have purchased a diffusion phantom as seen here. The aim of the project was to characterize the different properties of the phantom to help guide the development of a diffusion QA program here at NHS Tayside. This was done by determining the T1 and T2 of the 2 different compartments of the phantom as illustrated by the green and red boxes. Also, the ADC values of these compartments was acquired and compared to the values provided by the manufacturer. These are the graphs used to determine T1 and T2 of the compartments in the phantom. If you would like to know how this is done and find out more about our new diffusion phantom, please stop by my poster.